For lecture number 18 here on the reproductive system, we're going to continue talking about our developing embryo here, and we're actually going to back up a little bit and talk about some really early structures within the embryo. I do have to warn you, in chapter 28, your textbook provides a lot more details on embryonic and fetal development than what we're going to cover. Embryonic development is also known as embryogenesis, is a very complex topic. You can take entire courses on that in college, and we're just going to kind of hit some key highlights. So if you're reading along in the textbook, just keep that in mind. We are not covering the majority of the information presented on this topic. All right, so I mentioned in an earlier lecture there are 4 what are called extra embryonic membranes, which develop. Now these do develop from the conceptus. So the cells that are part of these membranes are cells that are derived from that very first cell of the zygote that forms when sperm meets egg. So they are produced from cells of the embryo, but they're not going to develop into the actual embryo itself and all of its little body parts. So there are 4 of these. One of them is called the amnion, and that's the one that's commonly called the amniotic sac. It's a transparent membrane, contains the amniotic fluid on the inside. And the purpose for that, if you're an embryo or a fetus, it's nice to have a buoyant environment for you to float around in. You don't have to worry about dehydration. It also cushions the developing embryo or the fetus if you're surrounded by fluid. That's a protective type thing. And it also helps maintain body temperature, since the amniotic fluid is going to take on the body temperature characteristics of the mom, and then that's going to transfer over to the developing embryo and the fetus. Where does the amniotic fluid come from? So interestingly enough, the fluid is actually derived from the mother's blood at first. It's produced from her blood vessels. And later, actually, the amniotic fluid comes from fetal urine. So the fetus does produce urine, the fetus urinates, but the urine just flows into the amniotic fluid as you might imagine. All right, another extra embryonic membrane is called the yolk sac, and it does kind of look like an egg yolk for a while. And interestingly, if you look at the early stage embryo of something like a chicken or a lizard inside an egg versus the early stages of an embryo in a human or another type of mammal, there are a lot of similarities. Very similar structures exist in all land animals. So a lot of these things do correspond, but they've taken on different functions for embryos that are developing in an egg outside of a body versus a mammal that's developing with a placenta inside the uterus of its mother. But just keep in mind there are a lot of similarities in these structures very early on between the among the embryos of different kinds of land animals. So the yolk sac is a sac. It's a little compartment on the ventral surface of the embryo, so the front side of what's ultimately going to become the anterior or the front side of the embryo. And eventually, the cells of this yolk sac are going to form part of the digestive tube. It also produces the earliest blood cells and vessels for the embryo. And it is the source of primordial germ cells. So the germ cells are cells that are eventually going to develop into the embryo itself. Then there's another extra embryonic membrane called the allantois, and it is at the caudal or the tail end of the yolk sac. It's a fairly small structure, and it becomes the structural base eventually for the umbilical cord, and interestingly, parts of it become part of the urinary bladder. So you have to remember, early on, these structures are composed of cells, and all of these cells in them contain the 46 chromosomes of this new individual. And you can think about those chromosomes, each chromosome as being like a recipe book. Genes are recipes, genetic recipes within that recipe book, they code for proteins that have many different jobs inside cells. But if you think about those chromosomes, so all these cells have the same sets of chromosomes, these recipe books, but different cell types, as you guys know, have to take on certain functions and certain jobs. And so, yes, some cells have to have many genetic recipes in common that they have to use for basic cell functions, but then if you're going to become a cell of the umbilical cord, you're going to read certain genetic recipes that are unique for an umbilical cord. Whereas if you're going to be a cell that's developing into a very early blood vessel, there are certain genetic recipes that have to be read in that cell type as well, so that it takes on those special functions. So that's why when you have something like the allantois, there are cells that make up this very early extraembryonic membrane, and some of them begin to read certain genetic recipes and become the umbilical cord, and others begin to read other genetic recipes and use those recipes and become parts of the urinary bladder. And all of these processes are very complex, and the nitty-gritty, gory details of how all this works are still not completely understood. All right, then the fourth extraembryonic membrane is the chorion. That is the outermost membrane surrounding the embryo, and that is the part of that we already talked about earlier. The chorion develops those little projections at the surface called the chorionic villi, and those dig into the endometrium of the uterus and help develop the placenta. All right, now how about the embryo itself? So at 17 days of development, so 17 days after conception has taken place, if you looked at the early developing embryo, it would look more or less like this. It wouldn't have these colors, but it would more or less look like this. So, and some of these pictures can be kind of confusing and hard to visualize, but this one is like you're looking down on top of a 17-day embryo. And then we're going to make a slice through it like they're showing you here on the diagram. And then if you cut this open and looked at it in a cross section from the side, this is what you would see. Okay, and so in right there, you have a layer of cells there that is developing into the amnion. That's going to be your amniotic membrane. And here's the very early amniotic cavity that's beginning to develop. Let me change pen colors there, right in here. That's your early amniotic cavity. And that's eventually going to expand and wrap itself completely around the embryo or the fetus, as we saw on earlier lecture pictures. And then down in here, this is your yolk sac, right in there. And interestingly enough, the very early embryo, this is what it looks like. Right there in the middle, that's it. It's called an embryonic disc. It's flat, and it has 3 layers. Let me erase what I've drawn already. So right in here, you have 3 layers of tissue there. And some of those layers blend into these other structures that we see here on the diagram. But I'm going to draw your attention to this top layer here called the ectoderm, and that's actually going to be over on the posterior side of our little disc-shaped flat embryo. If you were looking down on top of it, it would look more like that. And then the mesoderm, meso means middle, so those are going to be tissues that are in the middle of this flattened embryonic disc that's developing. And then right in here, this yellow line, which blends in with the yolk sac over here, this is called the endoderm. So these are your 3, what are called the 3 germ layers of the embryo, and not germ like a disease-causing microorganism. Germ here is referring to like germination. They're going to, these tissue layers are going to develop into various body structures. All right, so if you think back to this embryo, started out with one cell, one zygote. That cell has undergone mitosis many times, and various cells have taken on specialized functions. So some of those cells have become the amnion, some have become the yolk sac, and then some of them have now developed into the ectoderm, the mesoderm, and the endoderm. And then as these cells in here divide, they are going to go on to become various structures of the body. So they're going to become increasingly specialized. Okay, so this is a figure from your textbook which shows you, okay, so you've got these three layers in an embryo when it's in that disc form: ectoderm, mesoderm, and endoderm. And so the cells in the ectoderm, for example, continue to divide and take on specialized functions. So the cells that are part of the ectoderm, the easiest way to remember what they turn into is they become the skin and the central nervous system. And more specifics are given down in here about that, like the skin, the epidermis, hair, nails, brain and spinal cord, and so forth and so on. The endoderm over here, that inner lining, those cells go on to become your interior epithelial linings of the digestive and the respiratory tracts. So in general, those are the main things that cells of the ectoderm become and cells of the endoderm become. Now, once you remember that, everything else is derived from that middle layer. So those cells in the middle layer go on to become all of the other structures that you see listed here, such as your intervertebral discs, the walls of your digestive and respiratory tracts, not those inside linings, those mucosal membranes, those come from endoderm, but the actual walls themselves come from, the walls of those major organs come from mesoderm. Kidneys and gonads, connective tissues, your bones, your joints, your ligaments, skeletal muscles, almost all body parts internal body parts come from the mesoderm. So the easiest way to remember this is learn that the cells of the ectoderm become skin and CNS, cells of the endoderm become those inner epithelial linings, and then the mesoderm is ultimately responsible for all of these other various tissues that develop in the body. And why is that important? Well, think about, you've got this very early embryo. What if you have damage that occurs to the ectoderm? What types of tissues are ultimately going to be impacted? That could result in birth defects that impact the skin or the CNS. Or let's say you have damage to a very early embryo that occurs in the endoderm. So that embryo is likely or may possibly wind up with defects along the digestive or the respiratory lining. Damage that occurs to the mesoderm could impact any number of deeper organs and tissues inside the body, since so many things are derived from the mesoderm. This process by which an embryo develops into those three layers is called gastrulation. That's what that term is referring to. So you form the three layers. And then, as we've seen, that's going to set the stage for organogenesis. That means formation of all of the body's organs and systems. And we just reviewed, what key things are derived from each of those layers. Now, by the time you get to the 8th week, 8 weeks of development. So if you're thinking about gestation timeline, you're actually at 10 weeks of the pregnancy, or 10 weeks of gestation, because you got to add on those first 2 weeks before ovulation occurred. But by the time you've had 8 weeks after conception, all organ systems are recognizable, and that is the end of our embryonic period. The fetal period is weeks 9 through 38. And again, the numbers here, typically you think about a pregnancy lasting 40 weeks, but the fetus has actually only been developing for 38 weeks, approximately. Now, as you might imagine, this is a time of very rapid growth, lots of mitosis, takes lots of energy to create all these specialized organs and other structures within the developing embryo and the fetus. So that just gives you a slight taste of embryonic development and the development of those supporting membranes. And again, your textbook is going to provide a whole lot more example information if you're interested in reading more about the details of embryonic development. Lecture number 19, we're going to focus more on changes that are occurring in the mother during pregnancy, and that'll be the topic of our next video lecture.